Over the past 300 years, humans have studied cells, also known as the building blocks of life. Back with Robert Hooke, Rimac, Virchow, who first gave us the idea of cells, to the discovery of structures inside cells called organelles, to investigations into life cycles. We map proteins atom by atom, film the movement of molecules, so surely there are no rooms left in this house. Right? But recently, scientists realized something they'd overlooked. Not a molecule, not a protein, an entire organelle, the hemifusome. And this teeny tiny little structure is already changing how we understand cell communication, movement, and recycling. Before we get into that, a more well-known organelle must take the stage. To move cargo, get rid of waste, and just clean itself up a little, the cell uses a bubble-like organelle called the vesicle to go around like a garbage truck. But to do this, the vesicle must fuse to another structure, one with garbage or resources to move. This fusion is kind of like two soap bubbles coming together. They slowly start merging and conjoin into one. We knew it happened, but we couldn't figure out how. For years, researchers saw strange half-fused membranes and ignored them, assuming it was just a cellular glitch, a messy cell. They were blurry, in between, and easy to explain away. Until we asked a dangerous question. What if this wasn't a glitch? This fuzzy halfway point kept showing up. Scientists found that this movement was not just a blur, it was a controlled halfway state. It wasn't instant. The half-fused or hemifused process was careful. In a hemifusome, the two outer membrane layers merge, but the inner membranes are still separate. In this time, the cell pauses fusion and double, even triple checks that the two pieces are allowed to fuse. It's almost like unlocking a car without opening the door yet. The cell checks that this is the right place to fuse, that it's the right time, and the right cargo. Hemifusomes show that the cell is deliberate in its actions. They pause, think, and calculate, and only then will they allow fusion. This new process completely changed how we view membrane fusion. Instead of being messy and blurry, it was slow and regulated. We revealed a previously unknown process in vesicle fusion, and it has made us take a step back and look at our standard model of membrane fusion. If we were wrong about that, what other assumptions were we wrong about? Vesicle fusion also underlies neurotransmitter release, immune signaling, and waste removal. With our newfound understanding of vesicle fusion, we can now more easily detect and solve issues that impede these functions. It also tells us that we may have to reinterpret old data to see if this middle state was missed. And what if there are other secret structures out there, just waiting for us to find them in the shadows? For multiple centuries, we've studied cells, mapped them, named their structures, watched them work. It was so easy to believe that the age of new discovery was far behind us. But the hemifusome tells us that discovery is never over. Discovery is limited by how we look, by our perspective. This entire organelle went unnoticed, not because it was rare or hidden, but because it existed in a messy spot. And when we learned to look through the mess, at the secret control behind it, our understanding was changed. So next time we arrogantly think we know everything, the hemifusome reminds us, discovery isn't always on new horizons or in smaller and smaller places. Sometimes it's just about learning to see what is already there.